Hello everyone, my name is Ersin Atay. Today I would like to talk about how phosphate-solubilizing bacteria enhance phosphorus uptake and modulate the expression of the SPX gene family in apple. This topic is especially important for improving phosphorus use efficiency and sustainable apple production. Plant growth and development are largely regulated by 17 micronutrients, with phosphorus P being a critical macronutrient. While P is typically abundant in soils, only a minute fraction of it is in a dissolved form. This limited solubility contributes to substantial yield losses, and the continuous depletion of P in modern agriculture poses a significant threat to global food security. To enhance P uptake efficiency, it is essential to identify key root traits, genes, or beneficial microbial associations involved. Plants have developed strategies to facilitate phosphorus uptake and remobilization to cope with reduced availability. Plants undergo phenotypic changes to boost P uptake, such as developing a larger root system and excreting organic anion. P starvation also causes a wide range of molecular responses, with regulation occurring at both transcriptional and post-transcriptional levels. Although knowledge of P uptake and the P signaling system has increased recently at the molecular level, including protein-encoding genes and non-coding RNA genes, the molecular network underlying plant adaptation to P starvation is still to be elucidated. The SPX proteins, which contain only the conserved N-terminal hydrophilic SPX domain, are found in all vascular plants and could potentially serve as a new group of PI transporters. In plants, SPX domain containing proteins are classified into 4 subfamilies based on the presence of C-terminal extradomains. SPX members were first discovered in rice and Arabidopsis thaliana. A growing number of SPX genes have been discovered in plants, including wheat, soybean, tomato, and apple. MD-SPX2, MD-SPX4, and MD-SPX5 genes in apple were significantly expressed in response to PI deficiency. P deficiency affects around 50% of agricultural ecosystems worldwide and is a critical limiting factor for plant development. The use of P fertilizers has increased globally to address this issue, but 70% of the soluble P fertilizer in the soil rapidly combines with cations, turning into an insoluble form. The adaptation of plant roots to low dissolved P concentrations in the soil varies depending on the species and genotype. Plants with extensive root systems benefit more from P as they can spread over a wider soil area. However, characterized by a species-specific low rooting density, apples exhibit a limited capacity to effectively utilize P. P fertilization of apple trees improves vigor, flowering, yield, fruit quality, and storability. Therefore, enhancing the absorption and utilization of P by apple trees is of great importance, both ecologically and economically. Phosphate-solubilizing bacteria are a group of PGPR that dissolve unusable inorganic P in the soil, making it useful for plant growth. The primary hypothesis of this study suggests that enhancing the quality of apple nursery trees is achievable by reducing chemical phosphorus use by at least 50% in soils with sufficient P and incorporating PSP. To support this hypothesis, we identified SPX gene family members in apple and 4 other fruit species, unraveling the evolutionary history of SPX family genes through comprehensive analysis of phylogeny, conserved domains, selective pressures, syntenic relationships, and gene duplication events. Furthermore, our study delved into the expression patterns of SPX genes using transcriptome data from various apple tissues. We assessed the expression levels of these genes in apple plants, treated with chemical P fertilizer and PSP through quantitative real-time qRT-PCR analysis. Additionally, we sought to understand how the P nutrition levels of nursery trees treated with PSP would impact the quality of trees. Effective usage of phosphate-solubilizing bacteria is a promising tool to maintain the sustainable fertility of soil and improve plant growth. In this study, we applied half of the recommended P in soil analysis with PGPR. The results showed that the quality of apple nursery trees could be increased by reducing the use of chemical phosphorus by 50% in soils containing sufficient phosphorus, but by using appropriate phosphate-solubilizing bacteria. We supported these results by the fact that increased MD-SPX gene expression was effectively downregulated by increasing the level of available P in the apple nursery trees. Thank you very much for listening.